Probably the most important bit of respiration is the Krebs cycle, which directly follows the link reaction. And the reason why it's so important is because it produces the molecules that are involved in the electron transfer chain. So, if you remember from the previous video, acetyl-CoA is the molecule that's produced after the link reaction. So that enters the Krebs cycle. By the way, it might be important to mention where this Krebs cycle actually occurs. It occurs in the matrix of mitochondria. Okay? So, you guys remember the mitochondria, right? The mitochondria is the organelle, the aerobic organelle that kind of looks like this. And it's got his little, you know, foldy bits called cristae. And the center, the liquidy bit in the center is called the matrix, okay? And that's where the Krebs cycle occurs. Of course, what passes through into the Krebs cycle and into the link reaction is pyruvate. Glucose can't pass into mitochondria because they're too big. They don't fit in the pores, in the protein pores, or the protein channels. Anyway, okay. Now that we've mentioned that, acetyl-CoA pretty much joins onto another molecule. And this is this molecule over here, the oxalate 4C. Both the acetyl-CoA and the oxalate, or oxalic acid, oxaloacetate, I can't even pronounce it. Oxaloacetate binds to the acetyl-CoA to form this molecule, a citrate, which is a 6-carbon molecule. The 6-carbon molecule subsequently breaks down to a 5-carbon compound, an intermediate compound, and allows for the release of CO2 gas. Okay? Also, there will be removal of some hydrogen groups, and that involves the reduction of NAD to form reduced NAD. So you get NAD to reduced NAD. This 5-carbon compound will then be involved in the conversion of many different compounds. So first of all, there will be another release of carbon dioxide because it forms a 4C molecule, right? But also you'll get other processes. You'll get the conversion of FAD to FADH2. FAD is another type of enzyme, coenzyme at least, and it's involved in carrying this hydrogen group to the electron transfer chain, very similar to NAD. Again, you'll get 2 molecules of NAD being converted into reduced NAD as well. And also a very small amount of ATP will be formed from ADP. ADP plus an inorganic phosphate group will be involved in the conversion to ATP. So there's a very small energy yield, from this Krebs cycle. And of course, once the oxaloacetate is resynthesized, then the whole process can repeat, hence why it's called the Krebs cycle. The next part of this tutorial series will be looking at the involvement of FADH2 and NAD. Oh my god, I forgot, sorry, my bad. This is NADH, sorry. And yes, the involvement of FADH2 and NADH in the electron transfer chain. An important point to mention, by the way, is this ATP is formed by the direct addition of an inorganic phosphate group. And this is by a process of substrate level phosphorylation. Essentially, the ATP is formed by an enzyme reaction. It's direct. The ATP that's formed in the electron transfer chain, on the other hand, is done so by a process called oxidative phosphorylation, which is a very different process in itself. So we'll be reviewing that in the next video lesson.